Hi guys, today we are going to be talking about the exceptions to Mendelian genetics. So in the last lecture we talked about Mendelian genetics or complete genetics. And this is when we see dominant over recessive. Today we're going to be talking about the exceptions to Mendelian genetics. So the first one we're going to talk about is incomplete dominance. So we saw complete dominance, meaning that a dominant allele will mask a recessive allele. This is not the case in incomplete dominance. So what we'll see is a mixture of the alleles phenotype. So we see this happening in snapdragon. So we have a red snapdragon, a pink snapdragon, and a white snapdragon. So you can look at their phenotypes and their genotypes and see that when it is heterozygous, that it's a mixture between red and white. So in order for a snapdragon to express a white phenotype, it must have the two alleles that will code for white. And the same exact goes for red. In order for the red phenotype to be expressed, the genotype must be the two alleles that code for red. When we have an allele for red and an allele for white, they mix together to have a pink flowered plant. So this is incomplete. So incomplete means neither one is dominant and they will mix. So we are going to do an example. So we will mix a red snapdragon with a white flowered snapdragon. And we're going to set up our Punnett squares exactly the same way. Okay, so we are going to drop our red allele and our white allele, and we end up with all pink. Oh, wait, that's kind of messed up. Then we want to cross the F1 generation, and this is what we see. So the R is going to go down here, and then they kind of mix. It's a little bit off, so sorry about that. But you can see, again, when we have our two R alleles, it will have a red flowering snapdragon. If we have an allele for red and white, it will be incomplete, and we will have a pink flowered snapdragon. And then if we have the two alleles for white, then we will have a white colored snapdragon. Again, you can do the ratios. So 1 red, 2 pink, 1 white, and our genotypic ratios will match the phenotypic ratio. All right, so now let's talk about codominance. So codominance is when you have a heterozygous genotype. Both will be expressed. So they do not mix, they coexist, and so this is given the term codominance. So an example would be crossing a black chicken with a white chicken, and you see both of those traits expressed in the offspring's phenotype. So a black and white coexist in a checkered chicken. They're codominant. So we're going to do a Punnett square again. Okay, so we're going to cross a black with a white. We end up with a black and white, all four of them. Now we're going to take the offspring and we are going to cross them, that first F1 generation. And again, our phenotypic ratio, we're going to have black, checkered, or white. We're going to have 1 black, 2 checkered, 1 white. And our genotypic ratio, homozygous dominant, will be that, or I should say homozygous with the 2 dominant alleles, 1 heterozygous 2, and then those 2 white alleles is 1 as well. So they're very similar to, or the Punnett squares are very similar to what we did in the regular Mendelian genetics. It's just the way that they're expressed is different. Blood typing is kind of a mixture between codominant and recessive. So there are 4 blood types. They are A, B, or O, and and these are going to be the genotypes that code for the phenotype. So your blood type is actually the phenotype, what is expressed because of the genotype. So in the case of blood typing, A is dominant, okay, B is dominant. So when they're together, They're actually codominant. So people who have the genotype of AA, this would mean a homozygous blood type, they would have blood type A. If they are heterozygous, then they will still be A. Okay, so A is dominant over O. Same thing with type B blood. You can be homozygous for B, or you can be heterozygous and be type B. But if you have a type A with a type B, they're both expressed, and this one is codominant. And then again, you have O, which is your recessive. And the only way that type O blood will be expressed is if it's with another recessive O allele. So you can kind of see right here. So we're going to cross this. We have it in male with AB, we have the female with B, and this is how it's possible to get that AB blood type. Okay, and you have type B and type A. So parents that have type AB and B will have children 25% probability that they will be AB, 25% probability that they will be type A, or a 50% probability that they will be type B. The last type of, or not the last type of exception, but the last type of exception that you'll have to do Punnett squares are sex-linked or X-linked traits. Now, these are going to be linkage of disorders, diseases, or traits that are going to be linked to the sex chromosome, so either X or Y. Primarily the X, but you will see it on the Y as well. Some examples: color blindness, hemophilia, and baldness. These are all related to sex-linked traits. So the trait mostly occurs in males, and we'll talk about that in a minute. Females can be carriers of the trait, but they may not express the trait. And trait seems to jump generations because of this. So let's look at an example of a sex-linked trait. So the parental generation, you have your XY, which is going to be a male. He's normal. He's not carrying any genetic disorder within his sex chromosomes. Now we have a female. So remember, females are XX. She is carrying the trait that will have the disease. So we're going to set up the Punnett squares exactly the same as we would. So we have our female up top, our male right here. We'll let those go through. All right, so let's look at. So the F1 generation, 50% are females. Okay, that's pretty normal. If you have a 50% chance of having a female or 50% chance having a male. But now we're going to talk about it in terms of carrying that genetic disorder. So there's a 25% chance that that female will be a carrier. Now, why is she just a carrier and she doesn't express the trait or the disorder? And that's because she has another X. Okay, kind of masks this. So she's a normal functioning gene on her X chromosome that will take over for the non-functioning. Then we have the female who's normal. She's not carrying any trait. Then let's look at the males. So we have our XY and our XY. 50% of the male, in this case, normal. 50% are going to carry, or not carry, they're going to express the trait of color blindness. Now, why is that? Because they don't have another X chromosome with a functioning gene to help out this dysfunctional gene, okay? And so that's why mostly males are going to express it. So let's go ahead and cross again. So we're going to cross a normal female with a male who does have color blindness, and we're going to see how that generation turns out. All right, so we can see that 50% of the females, okay, or 50% are females, but 100% of them are carriers. 50% of, there's a 50% chance we'll have males, and both of them are normal. The last two types that we're going to talk about are called two types of genetic disorder, or genetic exceptions are pleiotropy and then also multiple alleles that will code for one trait. So pleiotropy is going to be a single gene that affects two or more traits. Okay, so one gene is going to affect two or more traits. So some examples in humans are sickle cell disease. So in sickle cell disease, it causes a misformation of the hemoglobin protein, but in turn, this also affects kidneys and other body systems. So that's why we say this one gene can affect many different traits or phenotypes in your body. And then the last one is polygenic. So polygenic are multiple alleles that are going to be coded by, or a gene that needs to code for multiple one trait. So height, weight, skin color, these are all going to be influenced by one or more gene. So in comparison, where we see monogenic, so traits are going to be determined by a single gene with two alleles, which we saw in Mendel's pea plants, that doesn't happen in polygenic traits. So more than one gene code for these. So height, weight, and skin color are all influenced by multiple genes, not just one. All right, so those are going to be the exceptions to the rule of Mendelian genetics, and you will have to practice codominance, incomplete dominance, and sex-linked traits using Punnett squares. If you guys have any questions, let me know and we will get it figured out. Thanks, guys.